We're continuing in this chapter with proteins. So proteins, they're involved in nearly every dynamic function in our body. They're very diverse. There's tens of thousands of different proteins that we find. They're each very different in what they do. In other words, in terms of their function, they're different, and their function is dictated by their structure. The amazing thing is that all these different proteins that we find in the body, they're made up of only 20 different types of amino acids. So another way to think about this is, if you think of the English alphabet, it has 26 letters, and from these 26 letters we create all these words. Similarly, think of these amino acids as letters, and with 20 amino acids we have all these different thousands and thousands of proteins that we find in living organisms. So amino acids, they're made up of an amino group. Now again, these amino acids are the building blocks of proteins, and they're made up of an amino group and also a carboxyl group, which makes it an acid. And in addition to these two, so both the amino group and the carboxyl group, they're bonded to a central carbon. In addition to the central carbon, we have what's called a chemical group, which is symbolized by R. And also there's a hydrogen atom that's bonded to the central carbon. So when you look at this slide over here, this is the central carbon, and attached to the central carbon on one end is an amino group. On the other end, we have this carboxyl group. You have the hydrogen atom here, and this is the chemical group that's represented by R. And this chemical group is what's going to make each one of these amino acids different. So amino acids are classified as either being hydrophobic or hydrophilic. So if you look over here at leucine, again, what do we have? You have the carboxyl group here, and this is the R group over here, and hold on, sorry. So yeah, you have the amino group over here. Now, same thing over here. You have the amino group, the carbon, the R group, the carboxyl group. Same thing over here as well. In leucine, what do we have? When you look at this R group here, this is different from that of serine, okay? And serine, the R group is different from that of leucine and aspartic acid over here. Now, if you look over here, this methyl groups here, this is hydrophobic. So because these molecules are hydrophobic, this whole amino acid is hydrophobic. On serine, you have the hydroxyl group. Same thing as aspartic acid, you have the hydroxyl group, in addition to the carbonyl group over here. So both these structures here, they're both hydrophilic. So again, both these amino acids are hydrophilic. The amino acids are essentially monomers that are linked together. And how do they come together? Well, again, through dehydration reactions. They come together and they form these peptide bonds, and they create longer chains, which we refer to as these polypeptide chains. Again, they happen through the same process by dehydration reactions. So again, when the amino acids come together, we end up creating these peptide bonds. Peptide bonds, they end up creating, the more peptide bonds you have, the bigger the chain. In other words, you have more and more amino acids, and you're creating a larger polypeptide chain, a longer polypeptide chain. So again, if you look at this amino acid over here, this amino acid over here, look at the carboxyl group of this amino acid and the amino group of this amino acid. What's going to happen is this hydroxyl and this hydrogen atom is going to go away. So what do? These guys go away to form H2O water, and then these two guys will link up with one another. So here we go. Through dehydration reaction, the OH and the hydrogen left. That's how you get the water, H2O. And then if you look over here, the central carbon now, this is this guy. This carbon over here, this number 2 carbon, is now what's linking this nitrogen group, okay? Of the second amino acid. So now we have 2 amino acids, so we have what we call this dipeptide chain. So probably the most important role for proteins is as an enzyme. This is probably one of the most important things in the human body that occurs, because enzymes, what they do is they speed up the rate of chemical reactions. In other words, they speed up the metabolic reactions that occur. So they serve as catalysts. So this regulates chemical reactions within a cell. It could be anything. So many different kinds of reactions occur. Again, they speed things up, slow things down because of enzymes. So other proteins are also important, aside from being enzymes. Some proteins, they're structural proteins. Again, for example, structural proteins like tendons and ligaments, right? Other proteins are contractile proteins like muscles. You have defense proteins. Antibodies are an example of defense proteins, which make up our immune system. Signal proteins, again, when you look at hormones and other chemical messengers, these are perfect examples of signal proteins. There's also receptor proteins. These guys, they transmit signals inside of a cell, giving the cell further direction of what to do. Transfer proteins also. Like hemoglobin, for example, is a transport protein. It's part of our red blood cell. This transports oxygen. Other transport proteins, they may be transporting vitamins, for example, vitamin A or thyroid hormones. Storage proteins serve as a source of amino acids for developing embryos. So if you look at an egg, you crack an egg open, you have the yellow part and you see the white part. The white part is the protein, actually, and that serves as energy storage, energy storage, protein storage source for that embryo to develop. So if you look at this figure over here, you have this looks like a mountain climber, and if you look at the hair, that's a type of protein. The nails, as this woman would have, that's another example of protein. Her muscles, the ligaments, tendons that she's using to climb, all are examples of proteins. So a protein-specific shape determines its function. So a polypeptide chain contains hundreds or thousands of amino acids that are linked together by peptide bonds. The amino acid sequence causes the polypeptide to assume a particular shape. So again, depending on which amino acids are there in which place, it's going to give that polypeptide chain a specific shape. So again, if you look at a protein and you see a figure which you're going to be seeing next, it might just look like a random ribbon, for example, but it's not. It's there for a reason. It's very, very specific in terms of the amino acids that are present, one after the other, after the other, after the other. So the shape of the protein determines its specific function. So again, when you look at this over here, this is not just random curling up of this polypeptide chain here. This is happening. Each one of these folds and the turns that this takes is there. It's done intentionally. In other words, it's because of specific amino acids that's present, it ends up taking these turns or goes straights. This example over here, again, it's the same the same protein that you're looking at in the previous example, but this one is showing a little bit more detail. You can see the carbon, where the carbon atoms are, the hydrogen atoms, the sulfur, the oxygen. So again, you get a more complete picture. This groove over here, for example, this is an example of the lysosome. So where this groove is how this protein will interact with its receptor. So for example, the bacteria, this protein will go attach itself to the bacteria at this end over here, at this part, at this groove. And this is how it's going to be able to function. So if a protein shape is altered, it can't do its job anymore. It can no longer function. So how does this happen? How can you make a protein not work anymore? Well, there's a few different ways, but let's go through the process of denaturation. So what ends up happening is, through denaturation, okay, the amino acid will start to unravel. In other words, it starts to lose its shape. Once it starts unraveling, it'll lose its shape. Once that happens, it loses its function. What can cause this to happen? Well, salt concentration, pH, or by heat. These three things that can change, it can break up, or in other words, denature a protein. A protein can have 4 levels of structure. You have the primary structure, the secondary, tertiary, and what's referred to as the quaternary. Some proteins do, others don't. So the primary structures of protein is unique. The primary structure of a protein is just the unique sequence of amino acids that you find within it. So the correct amino acid sequence is determined by the cell's genetic information. The slightest change in this sequence could affect the protein's ability to function. So for example, if you think about hemoglobin, it's supposed to be circular, or spherical in shape. Now some people that have this condition called sickle cell anemia, what ends up happening is that this red blood cell, it takes up a sickle shape, And this happens because of one amino acid that's out of sequence. Okay, that's wrong. It's not supposed to be there. It's there, and this ends up changing the entire sequence of this hemoglobin. The secondary structure results from coiling or folding of the polypeptide. So coiling results in a helical structure that's called an alpha helix. So a certain kind of folding leads to a structure called pleated sheets, which dominates usually you're going to find in the fibrous proteins that, for example, you're going to see in spider webs. This is an example of this fibrous protein. Coiling and unfolding are maintained by regularly shaped hydrogen bonds, okay, between hydrogen atoms and oxygen atoms along the backbone of the peptide chain. So look at an example over here. This is another example. This is an example of collagen. And again, what do we have? You have one polypeptide chain going over here. You got another one over there. You got another one here. So you got three polypeptide chains that are interacting with one another. This would be a good example of that quaternary structure. Okay, and we'll get to that in a minute. So the overall 3D shape of a polypeptide is called the tertiary structure. So the tertiary structure generally results from interactions between the R groups of the various amino acids. So what's there? Is that R group hydrophilic? Is it a hydrophobic? Okay, it's going to determine if that part of the protein, the sequence or the amino acid, the polypeptide sequence is facing out towards the water environment or inwards, for example. Disulfide bridges, they also help straighten that protein shape. So two or more polypeptide chains, okay, or two more polypeptide subunits, when they come together and they associate, they end up making what's this quaternary structure. So we looked at the collagen a couple of slides back. That's a very, very good example of a protein with a quaternary structure. So collagen has what we call a triple helix. Okay, it's three strands that are kind of twisted around one another. It makes it very, very strong in connective tissue. And we find connective tissue in bones and tendons and ligaments. These are three places. Well, there's other places we find connective tissue as well, but for now, we'll just leave it at that. So again, if you look at the primary structure, what do we have? Just a sequence of amino acids. Okay. So again, these guys here, these are all different amino acids. This is what we call the primary structure. Secondary structure, again, you start to see the shape that these amino acids take, okay, in connecting to one another. So this one, again, we call this the alpha helix, the way it's kind of twists around. And this one over here, this part over here is what we call these beta pleated sheets. So it kind of looks like if you look at it, another way to look at it instead of sheets, think of it, so we've got one sheet going here, one sheet here. You can also think of it this way, like there's steps, okay, coming towards the side. Now for both of these, what's keeping this sheet connected to this sheet are the hydrogen bonds that you find between these two sheets. So over here, between this oxygen molecule and this hydrogen, this oxygen, this hydrogen, this hydrogen, this oxygen, we have these hydrogen bonds, okay, and it's symbolized by these circles. And this one over here on this alpha helix, again, between this oxygen molecule here and the hydrogen molecule here, or this oxygen, this oxygen, this oxygen, this hydrogen, these circles represent the hydrogen bonds. This keeps this helical shape in place. Okay. The tertiary structure. If you look over here, this 3D, the entire 3D shape of this protein is called the tertiary structure. So this is the picture showing you of a thyroxine. This protein, here. There's transthyretin right over here. Okay, once it has what I said. So over here, this is the quaternary structure. Let's go back over here. So the quaternary structure of transthyretin. See one over here, another here, third one there, fourth one here. So these four structures, when they come together, okay, we refer to as the quaternary structure. This guy here, transthyretin, this protein, it'll transport vitamin A and also thyroid hormones. So again, another picture of a primary structure. It's just showing the sequence of amino acids. The secondary structure, okay, you can see the hydrogen bonds, and you can see the alpha helix and the beta pleated sheets. Sorry, going back here to the tertiary structure, you see the entire 3D shape of this polypeptide chain. And then the quaternary structure shows you all the four polypeptide chains interacting with one another and how they're put together to form this complete protein. Next part, we move on to our nucleic acids. So The amino acid sequence of a polypeptide is programmed by discrete unit of inheritance that we know as genes. Okay? Genes are made up of DNA, which stands for deoxyribonucleic acid. It's a type of a nucleic acid. DNA gets inherited from an organism's parents. So in the case of humans, we get one set of from our mother, another set from our father. DNA provides directions for its own replication. DNA programs the cell's activities by directing the synthesis of proteins. In other words, DNA has the entire blueprint for what a cell needs to do, what it needs to produce, how long it should take for something to do it, etc., etc., etc. It has all the information that's needed for that organism to function properly. DNA does not directly build proteins, okay? It's just the blueprint, it just has the information, the how-to. DNA works through an intermediary RNA, okay, which is for ribonucleic acid. So what ends up happening is this. In a nutshell, DNA, okay, will get transcribed into RNA. Right? Once DNA is transcribed into RNA, which occurs inside the nucleus, the RNA will then leave the nucleus, go into the cytosol of the cell, and then it gets translated. In other words, the RNA will get built into a protein. Okay? So if you look over here, this is a DNA, piece of DNA. And from this part to this part of this DNA molecule, there is this gene for something. Let's just say it's a gene for color, all right? And your body needs to produce color, melanin. So what's going to happen is this. This part of the DNA gets translated into RNA, okay, through a process that we call transcription. All right. Once the RNA is produced, again, this happens inside the nucleus of the cell. Okay. Once the RNA is produced, and by the way, these are both nucleic acids, DNA and RNA. What will happen next is that the RNA will leave the cell. Okay, the messenger RNA, it's actually called mRNA, will not leave the cell, will leave the nucleus and go into the cytosol of the cell, and there it's going to attach itself to a ribosome, and the ribosome will then start bringing all the different amino acids together. It's going to read this RNA molecule and start putting the amino acids together to build a protein. Okay, so again, in a nutshell, in the DNA, certain part of the DNA has a gene for something. In this example, we used to produce eye color. That part of the gene gets transcribed into RNA. Okay, both these things, they happen inside the nucleus of the cell. Then the RNA goes into the cytosol of the cell, where it gets translated and gets produced into a protein by putting the amino acids together. So DNA, again, stands for deoxyribonucleic acid, and RNA stands for ribonucleic acids. These are made up of monomers of nucleotides. So what are nucleotides? Well, nucleotides are made up of three parts. First of all, it has a 5-carbon sugar called ribose in RNA, and in DNA, it has a sugar called deoxyribose. Okay. In addition to that, it has a phosphate group and a nitrogenous base. So this is the phosphate group over here, this is the nitrogenous base over here, and this is the sugar over here. So the DNA's nitrogenous bases are only 4, right? And they are adenine, which is symbolized by A, thymine, which is symbolized by T, cytosine, which is symbolized by C, and guanine, which is symbolized by G. RNA also has adenine, cytosine, and guanine. However, it does not have thymine. Instead, it has uracil. So what do we have in RNA? We have adenine, cytosine, guanine, and uracil. So a nucleic acid polymer, a polynucleotide, forms from the nucleotide monomers. Okay? When the phosphate of one nucleotide bonds to the sugar of the next nucleotide, this also happens by dehydration reactions, and by producing a repeating sugar-phosphate backbone with the nitrogenous bases sticking out or protruding out. So if you look over here, what do we have? You have the sugar-phosphate backbone over here, and then you have these nucleotides that are sticking out over here. So here's that. The blue is here. Let's go start from the top. You got a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And what do we have here? The nucleotides that are sticking out. Adenine, thymine, cytosine, guanine, thymine. So two polynucleotide strands wrap around each other to form a DNA double helix. So the two strands that are associated because a particular bases always hydrogen bond to one another. Adenine always pairs up with thymine, cytosine always pairs up with guanine. Okay? And this is what we called base pairs. RNA is usually a single polynucleotide strand. Okay? So RNA is just by itself. DNA, again, you have a double helix here. So if you look over here, this is the double helix in DNA. And again, what do we have on this strand? Adenine, cytosine, cytosine. So what do you have on the other strand? A always pairs up with T, C always pairs up with G, C always pairs up with G. Let's continue. So T will pair up with A. Another way if you want to look at it, if you look over here at this adenine, notice it's like a triangle that's sticking out, and thymine, it's like a triangle with indentation. So this part is going to fit inside here. Same thing with the guanine. Guanine is kind of a circular part. Cytosine is kind of a caved in part. So this circular part will fit here. And this is just a graphical representation. So again, A always pairs up with T, C will always pair up with G. Okay, on one strand, so on one strand here, you have a C, on this strand here, you have a G. On this strand, you have a G, this one you have a C. So again, these are what we call these base pairs. So that pretty much wraps it up for this chapter. So the majority of people, they start producing enzyme lactase in early childhood. We discussed this earlier on in the beginning of the chapter. I kind of mentioned it briefly. Why? Because this happens as dairy products, they contain the sugar lactose. Okay, your body needs the enzyme lactase to break down that sugar lactose. So when your body is not producing enough lactose, or it stops producing lactose at all, then you have this problem of not being able to break it down. So then you have the problem, okay, of lactose lactose intolerance. So lactose tolerance, it represents it's a relatively recent mutation in the human genome. It's a survival advantage for human cultures with milk and dairy products available year-round. So those individuals, for example, the Europeans, Northern Europeans, again, they ended up having dairy products all year round. So what ended up happening is their body was able to keep producing this enzyme lactase. When you look at Northern Africans and the Asians, much hotter climate, long time ago, they were not refrigerators, all right? You cannot store milk or cheese and stuff, so they would be seasonal. And in that case, in those individuals, after childhood, the enzyme, the body started producing it, it would go away. Okay, so they lost that gene. But in the Europeans, because they kept on consuming it, they had a need for it. So their body, it kept that gene active for these European people to continue to produce the lactase. So they actually believe that the ability to digest lactose later on in life is actually a mutation. Okay, the normal thing they realize is that we should not be able to digest it. Okay, so another way to look at it. So research they identified 3 mutations that keep the lactase gene permanently turned on. The mutations they think that they occurred roughly 7,000 years ago. This kind of correlates at the same time cattle was domesticated in those regions. So after reading this chapter and studying it, you should be able to describe the importance of carbon in life's molecular diversity, describe the chemical groups that are important to life, explain how a cell can make a variety of large molecules from a small set of molecules, make sure you can go forward and backward, in other words, building things up, breaking things down, define monosaccharides, disaccharides, polysaccharides, explain what their functions are. Easy way to remember this, I think, would be think of examples of a monosaccharide, disaccharide, polysaccharide. Same goes for being able to define lipids, phospholipids, steroids. What are their functions? Where do we find them? Give examples. It'll be easy for you to remember. Describe the chemical reactions of proteins, their importance to cells. What do they do? Think about all the functions that we talked about in the lecture just now. Describe the chemical structure of the nucleic acid and how they relate to inheritance. Explain how lactose tolerance has evolved in humans. So again, kind of sum things up. If you have a short polymer and a monomer over here, you want to bring these two guys together, what do you have to do? dehydration. You want to get rid of the water. So OH from this part and H from this part is going to go away to form water, and you get this longer polymer here. Now, if you want to go back to this thing over here, what do? We add water, or hydrolysis. So when we add water, what's going to happen? The water is going to come, break up, or in this example, it's going to break the bond over here between these two guys, and hydrogen comes here, OH goes there. So again, you're back to what you started with. Okay, so that's what this arrow represents. It's a reversible reaction. Again, this bond stick model essentially shows you your carbon atom here, and it shows you the hydrogen atoms that are bonded to this carbon atom, and it's kind of showing you this 3D structure of these bonds and how they're laid out. Make sure you go through and answer all these questions at the end of the chapter that you find in your book. Again, same thing for these questions. Make sure you're able to answer them. You're able to do them comfortably. Most likely they will be, or some parts of it will be on an exam when you guys are being tested. Again, this is just sugar. It's just showing you two sugar monomers that come together to form this sucrose, two glucose molecules. Again, so this is your amino acid. Remember, don't forget the different parts of the amino acids. Remember what this group is called, what this group is called, what this part is called, right? And try to remember, will this be hydrophobic, hydrophilic? Probably for this chapter you don't know, but try to familiarize yourself with it for the chapters to come. Now this graph over here, this shows you the concept that for any given enzymes, okay, they're going to be different. And remember what we talked about, enzymes, they speed up chemical reactions, right? So one of the things that dictates the how fast a chemical reaction will go through is temperature. Generally speaking, for any given enzyme, there's an optimal temperature that the enzyme works best at. So an example of enzyme A, which is symbolized by this red curve over here, you see that right around, let's say 37 or 38 degrees centigrade, that the rate of the speed of the reaction is the fastest. Okay, this part represents the rate of the reaction. This represents temperature. This part. So over here, you have the reaction occurring the quickest over here. And then after this temperature, notice this is going to be the optimal temperature for the chemical reaction. Everything below that, it's speeding up. And then after this temperature, so after roughly 30 or 40 degrees, this 40 degree mark here, things start to slow down for enzyme A. Now going to enzyme B, what do we have? Is that we see that right around, so 40 degrees, things start to speed up. And roughly 78 degrees or so, or 79 degrees, this is the peak. This is what the chemical reaction is occurring the quickest, the fastest at. And then right around 80 degrees, things start to slow down. Okay, so yeah, temperature will affect the rate of a reaction of an enzyme.